Okay, wonderful. So we can still chat. We typically give a couple of minutes to to allow people join. So thank you again. I mean, joining us. I mean, not the convenient time, especially Dr. Liao, and this is my tremendous honor to have you.
Thank you, thank you. It's my pleasure too. You
Sure,
did a great service to the community. So
sure. So when is the best time to visit Taiwan?
Well, after the pandemic.
Yes, that's great. That's great answer. But any kind of season, fall or
Yeah,
spring?
I think avoid summer. Avoid summer is good. Summer is very hot. Yeah.
Very hot. Okay.
Yeah.
But probably similar to Korea. So Korea is
yeah. Summer is humid, hot, and warm. Winter is very nice.
Oh, winter is nice.
Very nice. Yeah.
So not cold.
Not cold at all. Yeah.
Oh, that's wonderful. I love nice weather. Especially I don't like the the cold weather, especially Boston winter. Horrible.
Yeah, winter is like California.
Oh, that's wonderful. That's wonderful. So that's probably perfect for you because you used to live in LA.
Yeah, that's right.
Yeah, I grew up in LA. I miss LA winters a lot.
Oh, so okay, that's interesting. So
you're from Terrell's group, right?
Yeah, that's right. I did my graduate work in Dave's lab.
Yeah.
So you grew up, California, and then you mostly done your degree in the California, and then you moved to the Boston.
Yeah, I moved to Boston, and then I got stuck.
That's interesting.
It happens. Look out.
You know what? I actually wanted to stay in California, but Chris Voigt actually moved from California to Boston, so I kind of a little bit stuck over there because I got my degree, in Boston, and then moved to there, and then came back. So that's interesting. Okay, I guess we probably want to start. Okay, so good morning, afternoon, evening. Hola, hello, bonjour, ni hao, konnichiwa, 안녕하세요. Hello, everyone. Welcome and thank you for joining us. The 9th seminar and Taiwan Day, or James Day. October may be the best month of the year with the clear sky, comfortable temperature, and crop harvest, and so on, for the most part of the world. At the same time, midterm exams would make students and professors very busy, including myself, and also other professors in this room. And life is sometimes full of irony or selection. When it is perfect to enjoy, we often do not have time. When we have a lot of time, we often do not have money or energy because we are too young or too old. So let's enjoy every single second of our times, not to waste or not to miss the beauty of the life. Okay, it's my tremendous honor to introduce very briefly our pioneer speaker, Dr. Jim Liao. I guess we don't need any introduction for him, but if I kind of try very briefly, he used to be a professor at UCLA and now the president of Academia Sinica in Taiwan. He is a member of NAS, NAE, and NAI, and all three of them. And many awards include an ENI Award for Renewable Energy and Presidential Green Chemistry Award from EPA, to name just a couple. He is joining us, I believe, his time at 11 p.m. So I'm really grateful for his kind presence and time from the other side of the globe. Dr. Liao, the virtual podium is all yours. Thank you.
Thank you, Dr. Moon. You did a great service to the community. Particularly, this forum is very helpful for the young people, and I admire your enthusiasm. And thank you for your wonderful service. And today, let me share my slide. Hopefully that will help people to understand and follow what I'm going to say a little bit better. Since this forum is for young people, so I assume that most people are probably too young to experience or even to learn the history of recent biology. If you study in the 80s or 90s, which I'm sure many of you have not even born yet, biology is a field to study natural organisms. And at that time, people tell you anything non-natural is called an artifact. So you don't want to do that. And the whole field is focusing on discovery of new molecules, new phenomena, new organisms that has already evolved. So if you say, I want to tinker with the biology system, you probably hear the comment, that's not biology. Okay. So, however, we recognize that even half centuries ago in 1960s, and that's way before probably some of your parents are born, people already redesigned biological molecules. The most famous one is LAC-Z. So LAC-Z practically defines molecular biology in early days, okay, until GFP was invented. And then GFP took over. Okay, those are reporter systems. And along with that, people discovered that you can make chimeric proteins. And again, that triggers many, many applications. Nevertheless, people don't view that as quote unquote real biology. Those are tools only, until people see that those quote unquote tools begin to win Nobel Prize left and right. Okay, and people begin to get more serious about redesign these biological systems. And beginning in 1990s, okay, a group of us start to engineer metabolic systems. Okay, that's probably one of the earliest examples of engineering more than one genes, a collection of genes that work in a system. So those, we call it metabolic engineering. And about 15 years ago, as you know, the term synthetic biology was coined, and that's created this field is still vibrantly working today. So the basic idea of synthetic biology, or what we call the design principle, is the following. Number one, biological parts are well defined, and that's generally true. As you'll hear from the following talk, the genetic code is always true, okay, across all kingdoms. Although you're going to hear that Dr. Deventer is going to change that. But by and large, the open reading frame is well defined. Promoters, sort of well defined. Enhancers, ribosome binding site, those are sort of well defined. Enzyme functions, people took that as a given. And then the second hypothesis was that biological parts are modularized and context independent. What does that mean? That means that you can move these biological parts across different species, even across kingdoms. And mechanisms are exact. People generally assume that once we know it, we know it. It's like mathematical rule, it's always true. And finally, people assume that effects are binary, plus, minus. That's how you learn in biology classes, biology 101, or even some of the graduate courses are still taught that way. You're activating or inhibiting. So these are quote unquote design principles. And these are what I regarded as hypotheses. meaning that they may or may not be true. And in fact, from my standpoint, these hypotheses are not really true. And most likely, for the most interesting problems, they are not true. Biological parts are not always defined, and they are not modularized. Okay. For example, enzyme functions. We thought enzyme function is just like what you learn in textbook. No, there are a lot of side reactions, a lot of non-specific substrates, and so on and so forth. And biological parts are not even always modularized. They are actually context independent. And those are probably the most interesting parts. So when people said that, oh, it's so easy to design things to move one part from one organism to another, and that's just in a textbook, that's just in class. When you really work on it, and for those of you who are still in graduate school, don't get frustrated if your project doesn't work. That's exactly what it is, okay? That's the fun part of synthetic biology. And even more so, mechanisms are not even always exact, okay? And we see examples after examples. For example, reaction mechanisms. And we thought that at least, come on, enzymes are enzymes, they catalyze A to B. And more, we found that in many cases, they characterize something called moonlighting functions, moonlighting the activities, complete different things. Or the mechanism can be slightly changed, the active site change slightly and catalyze additional reactions, not just a border substrate only. And this happens in metabolic pathways, and this also happens in signaling pathways. Finally, I think most of you, unless you are from computer science background, you'll probably know that the real world is not always digital. In many cases, the real world is analog. So we don't see always plus minus 0 or 1. Okay, we see a quantity. Okay, I think this concept probably is easier for synthetic biologists to grasp because we all believe in quantitation. And that is probably one of the strengths of synthetic biologists compared to the typical biologists, okay? Because we combine biology, chemistry, informatics, mathematics, even engineering. Okay? So in view of all these, I'm sure people will appreciate, okay, traditionally, we take evolved biological parts to redesign it, and we build, we test, we learn. This is a typical cycle that you hear so many times. And I like to stress that one of the key features of biology is that it is evolvable. And let's not forget this feature in biology, in synthetic biology. So that's the take-home message I'd like to give to people, that even though synthetic biology has made so many great strides, and the design principle in many cases are working reasonably well, but more and more, and in fact, more interesting parts are those that don't work. So in order to solve this problem, we need to study more and take care of more of the biological evolution aspect. Thank you very much. So that's a take-home message I'd like to leave people to think about.
Oh my goodness, thank you so much. I mean, for your insightful and timely and important perspective and vision, especially biology history. Wow, that is very insightful. I need to use your talk for my SynBYSS course today. Actually, in fact, I'm used many of your papers in my class. For example, I use articles on engineering promiscuous enzyme published in, I think, the PNAS and Ketchun's paper, etc. That's really great, the perspective and insight. Thank you so much. So, okay, so now the main speaker of today with a longer introduction, Dr. Jim Van Deventer completed undergraduate study at Stanford University and PhD study under the guidance of Dave Terrell at Caltech and postdoctoral study under the guidance of Dane Wittrop at MIT. That means he moved from California to Boston and then now stay at Tufts and then still, in the Cambridge and Boston area. He studied faculty position in chemical and biological engineering at Tufts University, as I said, in 2015, and currently holds the Bright Future Assistant Professorship. In 2019, James basically was awarded an R35 Outstanding Investigator Award for Early Career Researcher from NIH, and that's one of the great kind of honor given by the NIH. And 2020, last year, he was named an Emerging Investigator by the journal Molecular Systems Design and Engineering. And 2020, he was also named a Future Biot Leaders by the Division of Biochemical Technology of American Chemical Society. And the diverse members of his lab work at the interface of protein engineering, synthetic biology, and chemical biology. Primary interests include engineering yeast to better accommodate alternative genetic codes and devising new strategies to engineer more drug-like proteins. And the selection committee, do not have any kind of problem when select him as one of the speaker. And then because his resume and his achievement so far is so great. So Jim, take it away. It's all yours. Thank you.
Well, thank you very much. I really appreciate this opportunity. Let me just pull up my slides. So it's an honor to speak here today, and I really appreciated Professor Liao's introduction, in which he really emphasized the interplay between the design and the evolution within synthetic biology. There's really room for both in this area. And so what I'm going to tell you about today is some of our work in this area, where we're focused on manipulating the genetic code in yeast and figuring out what kinds of applications there are for manipulating the genetic code. So before I begin with the content, I want to acknowledge the people in the group who, as all of the trainees in the audience, I'm sure, are aware, it's a very difficult time to do scientific research with the pandemic. And so I am really, really appreciative of all of the work that everybody past and present in the lab is doing. So I just want to acknowledge them, and I'll try to point out them individually as the presentation progresses. So let's now go back to nature. And as Professor Liao mentioned, nature has really given us all kinds of amazing starting points for thinking about engineering function in biology. And yet, if you look at the way that proteins are synthesized, if you look at the way that nature synthesizes proteins, it uses the genetic code to do this. But if you look at the building blocks that make up proteins, then what you'll quickly see is that from a chemical perspective, this actually looks quite limiting. And so it turns out that nature also has recognized this over the years. And so nature actually uses a lot of post-translational modifications in order to engineer protein function in lots of different ways. And so like our hair has the properties that it has because of a whole lot of post-translational modifications, mostly of proline residues. So this is a very useful thing to note, and this is certainly something that in the laboratory that humans have started to do in very powerful ways. So there are good examples of taking proteins and doing post-translational modifications on them to make them more therapeutically relevant, such as the examples you can see here of protein pegylation, antibody drug conjugates, and even constrained peptides. However, there are a number of things that it would be really, really nice to be able to do with proteins and peptides that it's really, really challenging to do with only the canonical amino acids and with only the conventional genetic code. And so for that reason, a number of researchers over the years have thought very carefully about how they might genetically encode additional functionality within proteins. And we're in an exciting point in this field where it is possible to envision lots of different applications. So in my group, the kinds of work that we're really interested in doing is thinking about what are the therapeutic applications of non-canonical amino acids? How can we leverage the unique chemistries that we can find within these proteins? And what is it that we should be trying to do in terms of therapeutic discovery? And so some examples are shown here on this slide, and some more of our thinking can be found in this review that Arlinda, Mariha, and Manjie from my group wrote along with me that really sort of defines some of the problems that we're interested in. So to give you a little bit more sense for why we think this is an exciting area, if you look at proteins and small molecules, they really have complementary functions when they're used as therapeutics. And so small molecules can do really amazing things, such as inhibit enzymes, covalently interact with their targets, and even never let go in some cases. But there can be issues with specificity and off-target binding that happen with a number of cases. On the other hand, if we look at proteins, when used as therapeutics, can be exquisitely specific for their targets, but some of the other features that you find in small molecules, such as enzyme inhibition or covalent target engagement, those are basically absent from conventional proteins. And so why not try to put these things together? Why not integrate the small molecule functionality with the protein functionality in order to try and better access these complementary sets of properties? So we could do this with large small molecules, but really anything down to a couple of atoms change might give us something that's more therapeutically relevant than just a protein encoded by the conventional genetic code. So the vision that we have in the group then is to be able to discover proteins and hybrids that cannot be accessed using current discovery approaches. So really adding something to proteins with the small molecule functionality, and not just doing things that neither proteins or small molecules can do, but really thinking about when do these hybrids have advantages over other types of therapeutics, even very powerful cell-based therapeutics or nucleic acid-based therapeutics. We think that there's going to be nice times when these kinds of hybrids are very useful. So a grand challenge in this area is the challenge of how to engineer very specific inhibitors of things like metalloproteinases. And I'll just mention that as sort of the type of thing that we would like to do in the long run. So this is our vision. How is it that we're going to realize this vision? So this is where really the interplay between protein engineering, synthetic biology, and chemical biology comes into play, where in the group, what we do is we use genetic code manipulation to genetically encode non-canonical amino acids. And we're doing this in response to stop codons. And as alluded to previously, this is not something a cell necessarily likes to do. And so we have to think very carefully about how to do this and use both engineering and synthetic biology concepts to try and make this work better. But if we can make that work better, then what we can do is we can say, all right, let's use some powerful engineering tools for understanding the properties of these hybrid proteins that we're making, as well as for even thinking about how we might evolve or screen through those proteins in search of therapeutically relevant things. So that's really where we're headed as a group. So what I'm going to tell you about for the remainder of today is, first, I will tell you about some measurements that we have learned how to make in various ways to evaluate the performance of non-canonical amino acid incorporation machinery. So this includes the use of parts that other people have previously made. How well do they work in our hands? And then after that, I will briefly tell you some of the ways that we're thinking and beginning to apply the chemistries that we can incorporate into these proteins to do things that conventional proteins cannot do. And then in the last part of the talk, this is where I'll talk a lot about some evolution and high throughput screening that we're doing to try and enhance the ways that genetic code manipulation works in yeast. So those are the things I'll talk about today. I do want to briefly mention that we're also interested in things along the lines of intracellular delivery. It'd be really nice to be able to use proteins inside of cells routinely. And so that's something that Rebecca from the lab has started working on very carefully in collaboration with Xiaobing Zhu's lab. And another project that's ongoing that I would also be happy to take questions on afterwards is a yeast bacteria interaction system that Ming devised to allow us to interface yeast display directly with whole bacteria, which again has potential applications for targeting microbes in the long run. So let's get back to the main event, and let's start by talking about some measurements that we've made to try and understand how well genetic code manipulation works in our hands. And where we need to start from is the fact that cells did not evolve to work with alternative genetic codes. And when you tell a cell, hey cell, we're going to change around your genetic code, and we want you to start using a stop codon to mean something else, cells don't really like that. And in fact, cells are pretty good at fighting back when you start telling them to change around their genetic code. So a lot of times the termination machinery inside of a cell will actually truncate a protein that you wish you could genetically encode a non-canonical amino acid in. And so this means that if we're going to think about implementing these systems, we need to know how well these systems are doing in our hands. So this is something that we spent a lot of time doing. And we're not the only ones to do this. There's been plenty of groups who have thought about this question in various ways. And so if you look in the literature, you can actually find a lot of really creative solutions that people have found to try and enhance stop codon suppression. This is really true in E. coli. There's a few things in mammalian cells that have been quite nice. But in the opinion of my first graduate student, there is a lot of room for improvement when we move into yeast, which is where we're focusing our efforts. And so we should pause for a moment and say, wait a second, why bother to do this in yeast at all if there are really good tools in other organisms for doing this? Well, I gave you the reason of yeast display already, which is a really, really powerful protein engineering technique that has unique advantages. There's also a number of additional synthetic biology and metabolic engineering techniques in yeast that we think are going to be very useful for doing things in yeast in the long run. And there's also a number of resources in yeast that make it a really nice platform for doing engineering in general, that make it have advantages over these other organisms. But the disadvantage is we've got to do some legwork in order to try and figure out how is it that we're going to be able to do genetic code manipulation well in yeast over the long run. So our answer to how to make sure that we can do this well in the long run is we should be careful when we take measurements on genetic code manipulation systems. And so over the past few years, we've devised a few different reporter systems that allow us to be able to do this. Some of them are based on GFP and other fluorescent proteins. Some of them are based on a yeast display platform. And all of these systems allow us to evaluate how well protein translation is working when we try to repurpose a stop codon as a non-canonical amino acid incorporation site. So in order to make these measurements, we actually take a series of measurements. And the way in which we do this is described more in this work that I'm citing on this slide here. And this is also inspired very heavily by work that Jeff Barrick's lab did at the University of Texas. So the reason why we think this format is very powerful, as first demonstrated in E. coli by the Barrick lab and now demonstrated in yeast by us, we can devise a metric, and we can use this metric for telling us how well or how poorly protein translation is going. And basically what this metric tells us is if this metric is 0, everything is getting truncated in our system. Whereas if our metric approaches 1, that means that the stop codon suppression system we have is actually nearly as good as wild type protein translation. So this is a very useful metric for understanding how well our system is working. There's also a fidelity metric, which I will mention here and say, please take a look at the previous work if you'd like to learn more about that. So this metric, what does it give us? It gives us the opportunity to understand how well do the existing parts work for encoding non-canonical amino acids in yeast. And what you can see here is that when Jessica did these measurements using existing machinery from other groups, she did find some cases where things were working rather nicely, but she found some other cases where maybe that was not so much the case. But in any case, there's enough going on here that this really gave us an entry point for starting to think about how is it that we can start exploiting these functionalities right away. And so just to highlight that a little bit more, we can do what's known as click chemistry using several of the amino acids that we can genetically encode. And we can also even do some photo cross-linking using one of these amino acids as well. And so that's what I'm going to talk about in the next section of the talk briefly, before we get back into the question of how can we go even further with these reporter systems to try and enhance genetic code manipulation. So one more note before we summarize this section of the talk, and that is that we have a large number of additional reporter systems that each one of them has really nice properties under certain conditions. And with the range of reporters that we have, we can now basically evaluate genetic code manipulation in more or less any strain of yeast at this point, which is going to come into play at the end of the talk. So to summarize this section here, we can quantify non-canonical amino acid incorporation relative to wild type translation. And we've got multiple sets of reporter formats that are available for use in yeast, both in display and fluorescent protein formats. We've got access now already with existing machinery to a number of non-canonical amino acids with therapeutically relevant properties. And we continue to refine these tools so that we can use them in any scenario. So let's now turn and let's talk a little bit about where we're headed in terms of what is it we're going to do with these chemistries before we return to how can we better manipulate and enhance genetic code manipulation. So I'll tell you briefly about some of the things that we've done to exploit the kinds of chemistries. The first thing that I want to summarize is to say that one powerful way to apply these chemistries has been to generate some simple synthetic antibodies and then to systematically understand how it is we can put chemistry into these antibodies. So I'll mostly just describe these at a very high level, and Mariha and Haixing's very thorough studies here really go into a lot more detail as to some of the things that I'll briefly touch upon today. So the key questions that we wanted to ask in this work are, where should we put the chemistry after we can genetically encode it? And what should we actually do with that chemistry? And so to begin the answer, the question of where should we put the chemistry, we decided that we wanted to make a very simple synthetic antibody library that we could use in yeast display format. And so for those of you who think about antibodies a lot, you know that conventional antibodies have diversity all over the place. There's all sorts of different frameworks that contain various levels of diversity and all sorts of different loops called complementarity determining regions that also have substantial amounts of diversity. We did not want to have to address all of that diversity at once in the work that we're doing. We wanted to focus our efforts on how is it that we can combine antibody functionality with additional chemical functionality. And so we made this really simple synthetic antibody library in yeast format. And then we've been exploiting various elements of that library in various different ways. And so let me just tell you briefly about how we've been exploiting that library. So basically, with these very simplified antibodies, we can start to systematically ask, how is it that we should be incorporating different chemistries into these proteins? And what happens after we do? What properties are we adding to these proteins, and how reliably can we access them? So here's an example of the kind of data that we get out of doing these studies. So one of the nice things about having this set up in a yeast display format is that we can make all of our measurements for substitutions in these antibodies directly on the yeast surface without having to make any proteins solubly. And so what you can see here is data that Mariha took that allowed us to figure out, here is how well these different antibodies tolerate non-canonical amino acid incorporation at several different positions. And what you can see here is that in many cases, we're still observing binding, but the extent to which we observe binding has actually changed depending on the specifics of the situation. And we learned a lot more than that by doing some actual systematic titrations and things like that to learn more about binding affinities. But suffice it to say that we do have quite a number of functional chemically substituted clones that we have access to just with these systems here. And we have opportunities for improving them based on what we've learned from these initial studies as well. So moving beyond that, I will summarize in words that we can efficiently do conjugation in this format, and we can even do photo cross-linking in this format, and all of that has been described in this published work that you can read, where you can read more about it. But what I want to tell you now is, how is it that we can push beyond the model systems that we've been working with to more therapeutically relevant things? And to begin to answer that question, I want to say first that we're not just working with synthetic antibodies, we're also working with other types of proteins. And one type of protein we're working with are fibronectins. And these are very similar to antibodies in the sense that you can engineer them to be really high affinity for a given target of interest. So earlier this year, there was work out of Ben Hackle's lab at the University of Minnesota that described a really remarkable study where they actually showed that you could use thiol chemistry in combination with this warhead here to engineer fibronectins capable of interfering with human carbonic anhydrase function. So these are known therapeutic targets, and these particular conjugates have some really interesting specificity properties. And so Ben and I actually talked about this, and we said, hey, it would be really nice to see how far we can push the chemical aspect of these fibronectins. And so Mangier in my group decided, well, here's what we're going to do to begin to explore that. Let's systematically look at what are the different amino acids that we can substitute into a set of fibronectins from Ben's lab. What small molecule warheads that are known to target carbonic anhydrases can we present within, by conjugating these small molecules to the amino acids? And then which fibronectins do these kinds of conjugates actually result in functional hybrids in? And so we've got a lot of different hybrids that we can access using these kinds of systematic chemical manipulations here, both at the genetic code level as well as with the conjugation. And so to illustrate what we can then do is, because we are working on the yeast surface, we can ask all of these questions about the functionality of all of these hybrids at once. So this data here is data that Mangier acquired all on a single day, and after a couple of days of setup, and it gave us some really nice insights into what works well and what does not work well in terms of these conjugates. And so if you're looking really closely, you can see over here that these flow cytometry fuzzballs look a little bit different from all of the other flow cytometry plots that you're looking at on this particular slide here. So I'm not going to go into all of the details as to what exactly these mean here, except to say that these data indicate that when we incorporate this amino acid into fibronectins along with this warhead here, we get selective binding to carbonic anhydrase 9, which is not that different from what the previous work had shown with the thiol chemistry and this particular warhead here. So these two things really look nothing like one another in terms of chemical space. This is a totally different amino acid. This is a pretty different warhead from what was started with. And yet, we still see selective binding in each of those cases. And so what this tells me and what this tells our group is that we should be really excited about all of the different ways we can think about exploiting these kinds of chemical functionalities systematically and also in high throughput screening. And so to summarize this area, we now have a platform where we can start thinking about how to exploit the different chemistries that we can genetically encode in proteins. We can do this in synthetic antibodies. We can do rapid conjugations. We can do this in fibronectins, and the ongoing work in our group is really pushing this in a lot of different directions. So we're extending this to high throughput screening. We're extending this to additional protein scaffolds in a bit more rational approaches. We're also thinking about the different chemistries that we can exploit within these contexts. Okay, so we've talked about quantifying genetic code manipulation. We've talked about what are some of the things that we can begin to do with genetic code manipulation. And so finally, what we want to talk about now is how is it that we can push beyond what is already out there and try and devise better ways of accessing different types of chemistry, and even devising better ways of getting yeast to deal with the fact that we're trying to change around their genetic codes in really crazy ways. So remember, cells did not evolve to work with alternative genetic codes. They really don't like it when you try to make them do that. And so there are all kinds of different things, though, that we can think about doing to try and make it so that cells are better at working with alternative genetic codes. These are what I would call the usual suspects in terms of what types of things you might want to focus on engineering to try and enhance genetic code manipulation. But inside of a cell, there's all sorts of different components on top of that that might be relevant to enhancing genetic code manipulation. So let's talk a little bit about the usual suspects, and then let's revisit this concept of what else should we be manipulating to try and enhance genetic code manipulation in yeast. Okay, so the usual suspects, when you talk about genetic code manipulation, include amino-acyl tRNA synthetases. And these are really, really critical parts of adding an amino acid to the genetic code. And this is something that in my group that Jessica spent a lot of time figuring out how to engineer using the reporter systems that we discussed in the first part of our talk. So the end result of all of Jessica's work in this area is that we did a lot of directed evolution and high throughput screening to identify amino-acyl tRNA synthetases that had lots of different properties for doing genetic code manipulation in yeast. So we gained access to some new chemistries. We gained access to a wider range of specific and polyspecific amino-acyl tRNA synthetases. And we even have some pretty stringent ways of enhancing the functions of existing amino-acyl tRNA synthetases using the flow cytometry-based sorts that we're able to do with the kinds of reporter systems that Jessica described in the earlier part of her work. And so I wish I could tell you more about this work, but I can't really tell you too much more because there is quite a lot of it that just is not going to fit into the time of this presentation. But what I do want to show you is how we went from existing parts that other groups had engineered to actually engineering a whole lot of new machinery with some really interesting properties that moves beyond what the previously described parts can do in terms of in terms of genetic code manipulation. So, you know, in this work, Jessica has single-handedly doubled the number of known variants that are translationally active in yeast for genetic code manipulation. And there's all sorts of other good things in this work that I would love for all of you to read and give us feedback on as we continue to work on it and work in this area in general. So that's the usual suspects. let's now talk about what more should we be doing to try and enhance genetic code manipulation in yeast. And so rather than just think about these usual suspects, a few years ago, we started thinking about in the group, okay, what kind of genes might we want to knock out to try and enhance genetic code manipulation? And so the simple way of addressing this question was to take some known knockouts that are involved in termination of translation and in translation in general, and start to run some assays to see if those knockouts would actually enhance genetic code manipulation. And so borrowing from our reporter systems that we talked about in the first part of the talk, you can see here that the very classic yeast strain BY4741 exhibits this level of read-through efficiency under these conditions here. But if we actually take a knockout of that strain, the Delta PPQ1 strain, then what you can see here is a substantially enhanced efficiency of genetic code manipulation. There also seem to be enhancements in some of the other strains that we've looked at as well. And we also looked at these measurements under enough different conditions that we're pretty convinced that these measurements actually represent biological changes that are happening and not just artifacts due to the kinds of reporters that we're using here. So this was pretty interesting in its own right. So to take this a little bit further, we said, well, you know, we should make sure that we can actually do some model enrichments comparing these two strains and see if we can pull out the strain that seems to exhibit an enhanced phenotype. So we took even more of our reporter systems and we did some model enrichments between the single gene Delta PPQ1 deletion strain versus its parent strain. And you can see here in flow cytometry format that after enrichment, it looks like we were observing enhanced amounts of stop codon read-through based on the fact that these red sorted populations are starting to peak out above what the unsorted populations look like. This has been quantified here, where yes, indeed, the amount of cells that exhibit high performance phenotypes seem to be much higher. Of course, this was a model sort though, where we were only looking at two strains here. So yes, it does look like we're able to go to a sorter and enrich for the phenotype of non-canonical amino acid incorporation, all very encouraging, but how far can we push this? And so ongoing work in the lab right now is work where we are actually looking through an entire 6,000 strain knockout collection to try and see what strains enable us to enhance genetic code manipulation at sort of the genome-wide level. And so thanks to Maitreya Dunham's lab at the University of Washington, we received a pooled collection, and Matt, who is an undergraduate in the lab, working with Jessica, transformed this collection with a series of reporters and then did a series of screens to try and figure out, are there any strains in here that will enhance genetic code manipulation? And so we're in the middle of this phase of the work here, where in fact, we've done most of the identification of the different strains that have come out of this screen. And we've started doing some validations to understand what are the properties of some of these things that are coming out. So here, very briefly, is roughly what the screens look like, where we start with an unsorted population shown in red here. And of course, we've got our reporter systems in every single cell that's coming through these screens. And so gradually, as we are stringent in the way that we're looking for the phenotype of non-canonical amino acid incorporation efficiency, we're able to isolate populations like this one here, this orange population that exhibits a pretty substantially enhanced read-through efficiency performance level in comparison to the unsorted population. And when Matt has diligently looked through all of the 12 tracks that he sorted, he actually identified 55 candidate strains that may enhance genetic code manipulation. And he's even been verifying some of the lead candidates. And so let's talk just a little bit more about what it is that we're finding so far. So let's tell you at a high level, here is what we have found. And what we have found is that the vast majority of strains that are identified in the screen have no known connection to DNA and protein translation. It's just not known based on what we know about the yeast genome so far, according to the Saccharomyces genome database. So that is really surprising. And so you might be asking, okay, so like, are these hits for real then? And the short answer to that question is well, we think so, at least in the case of a couple of lead candidates that we've been characterizing in a lot more detail. And so we've acquired a number of strains from independent sources that have the same knockouts that we identified in the screen in order to try and make sure that what we're observing in the screens is something that transfers over to other genetic knockouts. And the short summary of what Matt has seen is that indeed, it looks like some of these strains, two of them in particular that we've validated very carefully, are strains that do actually support enhanced genetic code manipulation in comparison to the parent strain. And let me just emphasize that these two strains that I'm showing you data for here are strains with no known connections to protein translation whatsoever. So we think that this is really exciting in terms of how we can enhance genetic code manipulation for the future. And so let me wrap up this work in this area as follows. So we think that there's a lot of different opportunities to improve genetic code manipulation in yeast. So very clearly, Jessica's work has shown that we can engineer aminoacyl tRNA synthetases in all sorts of different ways. We can also, though, look at knockout collections and systematically look through what happens when we start knocking out different genes from the genome. And so what this means is that there's all sorts of other things that we can think about engineering to try and enhance non-canonical amino acid incorporation in yeast. And really, like in terms of genomic diversity, I would say that anything is on the table at this point. You know, there's all sorts of really smart people who have thought about how to engineer cell strain performance to be much better than what it is, but not many people have really thought about this in the context of genetic code manipulation. And so we think that there's a lot of opportunities for taking the tools from metabolic engineering and from some other synthetic biology work that we think is going to enable us to really push things in lots of different directions. And this, of course, includes things like the SC 2.0 project, where literally there are yeast that have partially recoded genomes, and that project is on the way to having a completely recoded genome where there is no more amber stop codon. So that, I think, is a very exciting thing that we hope to be able to take advantage of in the future. And then finally, I think that we're going to learn something about biology by doing all of this engineering in the long run as well. So just to wrap things up at the higher level, I told you today about measurement. Then I told you about how we're beginning to exploit chemistries that we can genetically encode. And then at the end of the talk, I said, here is how we're starting to expand genetic code manipulation beyond what we have access to right now. And we're really excited about all of these areas for the future. And all of this work is the result of some very dedicated trainees doing some really excellent work, everybody from undergraduates to postdocs. So I'd like to thank all of them and thank the funding sources, as well as you for all of your time and attention. And at this point, I will stop talking and take your questions.
Wonderful. That's fantastic, Jim. And this is, of course, a very challenging test because you need to fight against mutations and evolutions. So any question for him? Let me check. I mean, whether we have any question. Okay, so in that case, while waiting, oh, we got the question. Actually, I'm going to ask. Okay, so we have the question. So from Matt, do you have any hypothesis on why the lead candidate with no known role in protein synthesis in the pooled knockout library affect NCAA incorporation efficiency? and then I wait, your answer, and then I will ask the second question later.
Yeah. Okay. So, so I guess the way, so in terms of specific mechanisms and specific hypotheses in terms of here is the mechanism by which the particular knockouts we found seem to enhance non-canonical amino acid incorporation efficiency. We don't know the answer to those questions at the moment. However, if you think about protein translation is a very complicated process. And you can imagine that a lot of aspects of a cell, including things like metabolism and other things, like they're all in some way or another related to protein translation. I would argue that, the network that protein translation touches is actually quite broad within the cell. And so my hypothesis is that there are more things that are connected to protein translation than what we've been able to directly link based on the existing biological studies that are out there. And so that's one of the reasons why we think this is a very powerful tool for biological discovery as well as for engineering, is we might be able to take some of these observations that we have ongoing and then sort of look backwards and try to figure out what is the biology that underlies that, so that we have a better sense for more specifically why it is that, like a knockout in a metabolic gene might be affecting protein translation. But that's a really important question.
Sure. So second question from him again is, how many Ncaas have you been able to incorporate into a single protein? And can you think of therapeutic modalities where more than one would be useful?
Yeah, so those are great questions as well. So in terms of the work that we have done in the lab, the most that we've investigated at this point is 2 stop codon suppression events in one gene where we're trying to genetically encode the same amino acid, or at least that's the most that we've reported at this point here. We are thinking about that pretty carefully. But at the present, if you look at the data that we have at this point, you can see that we're still relatively inefficient in yeast in terms of stop codon suppression. There are some really nice systems in E. coli involving recoded E. coli plus a combination of a number of other things that can get you quite a few stop codon suppression events in a single gene or in a single protein. So there are some examples of that are out there. There's also different non-canonical amino acids that have been genetically encoded within the same protein. In terms of the question of like, how would you want to think about trying to use more than one non-canonical amino acid in a therapeutic setting? So one thing to think about is, could you have 2 different amino acids that are doing 2 different things that are both enhancing the therapeutic relevance of a protein? For example, it could be the case, I mentioned the idea of covalent target engagement, like an amino acid that's really good at doing something like that might not be very good at targeting an enzyme active site. And so you could think, well, maybe there's a way that you could try to do both at the same time, but maybe you would need more than one functionality to do that. So that's the kind of thing that you could think about in terms of more than one example. Or
that's
more than
wonderful.
non-canonical amino acid. Sorry.
Yeah, that's wonderful. Okay, so I just want to ask one tough question to you, because I always kind of wonder, what would be your perspective on evolution or mutation, or the why we have the protein in that way. So my question is, why do we have current redundancy in terms of amino acid coding, and why not more amino acid or fewer amino acid in protein, but just 20 or 21 if we include selenocysteine, or 22 if we also include pyrrolysine, I guess.
Yeah.
Do you have any perspective on that?
Yeah, so that's a really good question. Okay, so why don't, in terms of the question of why don't we have more amino acids in the genetic code? I think a short answer to that question is that nature has been really clever about using post-translational modifications in really, really powerful ways. And so I think that can compensate for the fact that there's not more amino acids in the genetic code in some pretty surprisingly powerful ways. In terms of why is it that we have such degeneracy in the genetic code? I do think that in terms of the way that proteins evolve and the kinds of things that happen when you have to replicate DNA over and over and over again, I think that having some amount of robustness and some amount of automatic error correction, where a mutation in the DNA is not necessarily going to change the meaning of a protein translation, I think that can be quite good in terms of making sure that UV irradiation or other sorts of DNA mutagens are going to immediately just tank the fitness of an organism or the function of a protein. I think that those kinds of things are probably some of the likely reasons why there isn't more degeneracy in the genetic code. One thing that's going to be really interesting, so there's now somewhere in laboratories on this planet, there are E. coli strains that have only 63 codons. There are E. coli strains that have been publicly reported that have 61 codons. And George Church's lab has reported that they continue to work on a 57 codon strain of E. coli. So what's gonna happen when those strains start to evolve? I think that there's going to be a lot of interesting things we might learn about codon degeneracy and other related things when people do more and more experiments with those. And some of those things are ongoing already.
Sure. That's why I asked that question, because of the currently people kind of working on those kind of project to answer probably my question or their question. Okay, so we now past the 11 my time. So okay, thank all for joining and staying today again. And next week we will have Professor Ling-Chung Liu from Duke University and Professor Xiao Jun Tian from ASU. See you next week, the same day and time, and the follow-up Zoom chat will occur via the link in the chat box. And basically you could just click that one if you wanted to join us. And see you all soon in a minute. And then we'll see you over there. And then James, please click that button, and then you will automatically move to the other Zoom.
Awesome, thank you.
Okay, thank you so much. Wonderful talk. Thank you.
Thank you.
So I will click this one as well. Okay. So Tony, you could now close and then stop recording.
Thank you.
Thank you so much. Have a nice day.
You too. Bye-bye now.
Thank you.
Thank you as well.
Thank you.